All right, this is the CAM project. I'm Tyson Kaufman. I'm a student athlete here at Concordia, majoring in biology. I'm a junior, and following graduation from CSP, I intend to attend medical school. And like one of the reasons why I really enjoy research and why I decided to go into research is I wanted to be able to enhance and develop my critical reasoning skills. So it gives me an opportunity to learn and build skills outside of classroom setting in more of an uncontrolled environment, working with trial and error and like developing hypothesis, testing the hypothesis, and so on and so forth.
I'm Margot Coombs. I'm also a biology major. I'm a junior, and I also plan to go to medical school. And I joined research just to get experience testing original ideas that our group has come up with, and just gain more realistic lab experience outside of, just running through a lab manual and those type of protocols.
I'm Samantha Cole. I am also a biology major and a junior. My plan is to go to physical therapy school after this. And one of the reasons that I decided to do research was just because it allows you to kind of use your creative side. So obviously you have to problem solve a lot, but you can be creative about that. And again, just like apply real world skills in a more realistic setting.
And I'm Kayla Renwick. I'm also a biology major and a junior. And then after CSP, I plan to go to physical therapy school. I really like research and wanted to be a part of this research project just because we don't know what we're going to discover while we're going through it. So we're learning as we go along with Dr. Yang. So that's just interesting and a different setup than other classes are on campus. But then it's also been a really good chance to gain lab experience while still in a learning setting. So we can kind of learn and make mistakes as we go and have it not. So it's just a good setting to learn lab technique and everything like that. So then before we get into the bulk of the presentation, we just want to go over some terminology quick. So this, so In-Ovo, so that's when like a chick developing inside its eggshell. So just like a normal chick that would grow and then hatch. Ex-Ovo, so that's mostly what we work with. So this is chick developing outside of the eggshell. So those two images over there on the right, like the top one and then the one that's in the middle, those both show ex-Ovo embryos. And then CAM, which we'll be referring to a little bit throughout the presentation, is a chorioallantoic membrane. So that's the external vasculature of the tick. So in those two images, it's the red vessels that you can see all around in the yolk. And then lastly is the THP, or truncated hexagonal pyramid. This is the image on the bottom over there, and it's a vessel that was developed by a past research student. So we just think that's pretty cool that somebody that was in our project at CSP designed and developed this vessel that we have then used throughout the project to house our ex-Ovo embryos.
Okay, so just kind of looking at our project overall, we have been working on growing chicks ex-Ovo. So we've grown them in a couple different types of vessels. We've grown them in a cup, we've grown them in a weigh boat, a cube, our THP vessel, and more recently in a tumbler cup and a shot glass. And basically what we're doing is we're looking to study the CAM, which is in this left picture right here. It's the blood vessels that are over the developing embryo, and we want to be able to vascularize a tissue scaffold, and this will go into like tissue engineering hopefully in the future. And we're also just looking at different causes of ventral body wall deformation that we'll get into in a few slides. We want to know if this deformation is avoidable, if it's inducible. But overall, looking at this project, we want to be able to basically take a biopsy. We want to take a piece of healthy tissue from the body, and then we'll isolate that healthy cell. We'll cultivate it, we'll proliferate it, and then we want to be able to add it to a scaffold and grow that tissue into an organ that is needed for a person that is like going through organ failure or needs a tissue replacement, something like along those lines. But the problem with current research is that these scaffolds aren't vascularized. So when they're put into the body, they're going from an oxygen-rich environment to a hypoxic environment. And this is eventually preventing them from growing and developing with the body. And they tend to just die when they're cut off from their oxygen source. So we want to be able to vascularize our tissue scaffolds so that when they do go into the body, they will still have a tissue and oxygen and nutrient supply so they can continue growing, and they won't just die as soon as they're implanted into the body.
Yeah, and then our project as of right now is kind of taking two routes. The first route of our project is we're looking to create or even find a vessel which optimizes the chick's growth, viability, as well as the visibility of the cam, which is that vasculature which Kayla had talked about prior. And then we are looking to optimize the visibility due to our use of it within tissue engineering. Some of the different vessels which we have used are shown there, such as like the cup, which has the saran wrap type film to nest the egg, weigh boat, which is just your common weigh boat, which is used within labs, and then two different vessels which optimize a siding that allows for oxygen diffusion across its membrane. And then right there on the right is our truncated hexagonal pyramid, which we have put together. And then the second route of our project is we're looking to what is the root cause of that ventral body wall deformity being seen. So chick A there has the presence of that ventral body wall deformity that is showing a complete ventral body wall deformity. And then chick B is a chick which is grown and hatched, or not hatched, but was grown In-Ovo, so within the eggshell. And there's clearly not that ventral body wall deformity present. So we're looking into different things which may be the root cause of that. We have looked into things such as ethanol exposure, vessel type, turning, oxygen supplementation, and this is stuff that Sam will talk about in a little bit as to what we have hypothesized.
Yeah, so kind of getting into the cracking process. So this first video, we'll get into cracking it. So first, we'll take the egg outside of the incubator and then use the wide end to turn it up so the wide end is facing to the top. And that kind of has the air pocket. So we're making sure that we're not going to rupture the egg or the embryo that's inside. And we will poke a hole at the top and then turn the egg to the side, allowing the egg to float to the top so that when we take 10 to 15 microliters of the albumin, that again is not ruptured. And so you can kind of see that process going right there. And then after we complete that, making sure that the egg shell is smooth on the edge, you'll smoothly transfer it into the vessel, whatever vessel we choose to transfer it into. And so this is probably the hardest part for sure of this whole process, because you kind of just have to go for it. It's not really something, that just got to go for it.
There we go. Okay. So after you transfer the vessel, so something that, or transfer the embryo, sorry, something interesting that we've been seeing with some of our smaller vessels that we've used, so this is like the small cube and the shot glass, the embryo will flip from the bottom up to the top. So it'll just kind of float to the top due to the buoyancy of the yolk. So in this video, if you watch on the right side, you can see the little red embryo, and it floats from the bottom up into the top. So we want the embryo to end up on the top like that, because if it stays on the bottom, then the embryo suffocates and eventually dies. So this is something that we've kind of noticed with the smaller vessels, like I said, but then the larger vessels don't really do this. So then stuff like the Weibo and the tumbler, and even the larger cube, they don't do this. So then in order to get the embryo to flip up to the top, we have to either transfer it again from the vessel that we just transferred it into a different one, or we have to manually flip it. So we take a little piece of the eggshell and we like pull it across the yolk to try and get the embryo to flip up to the top. But this then puts the embryo at a greater risk of rupturing, which is then also going to decrease its ability to survive. So having it flip like is seen in the video is ideal, because then it's less chance of it rupturing again.
All right, so one thing that we've been playing around with a bit is our In-Ovo incubation time. So when we're getting ready for a crack, we actually incubate our chicks in the actual shell before we end up transferring them. And this just helps them like grow a bit stronger, and then we're actually able to see if their hearts are beating and we can tell if they're alive. So for in previous semesters, we've always incubated for 55 hours, and we haven't really changed that up much until last semester when we started incubating for 72 hours. And we did this in hopes that the chicks would actually be closing their ventral walls while they're in the shell, and that the chicks would be stronger and easier to transfer. A lot of times it would be really hard to tell if the chicks were even alive because the embryos were so small. And they don't tend to survive the transfer process very well. So we were seeing pretty low transfer viability. So once we started incubating for 72 hours, we were seeing a higher transfer viability rate, but we were also seeing that the embryos were rupturing more, so their egg yolks would break and start spilling outside of the, spilling in the ex-ovo vessels. And we think that this is because the vitelline membrane, which you can kind of see in that lower right picture, is starting to fuse to the actual eggshell, making the transfer process a little bit more difficult. So next semester, we're going to try incubating for 65 hours, and we're hoping that this will be a sweet spot, that the chicks will still be strong enough to handle the transfer process a little bit better, but they won't be sticking to the eggshell and rupturing.
All right, so then what is the ventral body wall deformity that we're talking about? A ventral body wall deformation is a failure of the midline to close. It's that ventral midline, the abdominal thoracic region is not closing. So it is seen as being as an opening. There are different types of ventral body wall deformities that we have noted. An upper ventral body wall deformity, what you're seeing on the left there, it is occurring when the heart is observed outside the body. And then the opposite would be when a lower ventral body wall deformity. And in this one, you are seeing the intestines outside the body. That one is seen within the middle there. And then another possibility for the deformation is that for both the heart and the intestines are being located outside of the body. And this one referred to as a full ventral body wall deformity. And that is the picture on the right where you're seeing both the heart and upper thoracic region as well as the abdominal region being open.
So going into our hypotheses, we kind of have three that we have developed and tested throughout these past semesters. And so the first one being that ethanol was inducing the ventral body wall that was being seen. And this hypothesis was kind of looked into because of a human condition called fetal alcohol syndrome. And so we thought that that was kind of a lot like our ventral body wall. The second one was that the large circumferential volumetric space was causing the ventral body wall deformation. So obviously our vessels are a little bit bigger than the eggshell that they're being grown into. And so that's putting stress on the developing embryo. And then finally, hypothesis 3 is that turning the embryos in ex-ovo vessels will decrease the ventral body wall deformation. And so we just thought that chicks turn during their regular development when they're growing in ovo. So we thought turning would distribute egg contents evenly, decreasing the ventral body wall rates. And so we'll kind of look into this in these next few slides.
All right, so getting right into hypothesis 1. Again, we were hypothesizing that the 70% ethanol, which was used to sterilize the vessel, is what was causing the ventral body wall deformity. And that was in comparison to, again, the human model, as in the fetal alcohol syndrome. So we were predicting that that was causing the failure in ventral, the midline closure that we were seeing. And to test the hypothesis, our experiment was we had 3 varying ethanol treatments. So we treated chicks with 10, 20, and 30% ethanol. And then we had control chicks as well, which had no ethanol treatment. And what we would do is we would crack into the vessel, and then we would add 250 microliters of each treatment. The control would just receive distilled water. And what you're seeing here within this figure is that the ventral body wall deformation was still being seen as present within the ethanol as well as the control. Panel A is an embryonic day 14 chick, and it is not induced with ethanol grown chicks. With an ethanol grown chick, as well as panel B and C, they are embryonic day 16, and they are control chicks as well. And in all 3, a clear ventral body wall deformity is being seen. So then going on with the hypothesis, again, we tested ethanol versus control treatments, and the result is being seen here in the graph on the right. Ventral body wall deformity was being seen within treatment and control chicks. So the first bar here, the graph, you have no ethanol treatment, so this is going to be the control chicks, which have no treatment of ethanol, and we are still seeing a ventral body wall deformity within 8 of the 19 chicks, which were grown ex ovo. And then the other 3 here, the wayboat, the THP, and the cube, we are still seeing ventral body wall deformity within those as well. And then the significance here is that on the far right, the bar, these are chicks which were hatched in ovo, or I should say grown in ovo. They have the presence of no ventral body wall deformity, which is why we are saying that this is a problem. So in conclusion, we found that ventral body wall deformity is independent of ethanol sterilization techniques. So leading to our next hypothesis, we decided to test the effects that large central and the volumetric space has on ventral body wall deformity.
All right, so as Sam kind of talked about earlier, our second hypothesis was that large circumferential and volumetric space are causing this ventral body wall deformation that we're seeing. We think this is because the vessels that we're using aren't applying the needed pressure or physical constraints to help the embryo close its that ventral midline. So what we tested was a bunch of different vessels of different sizes. So we tested some vessels that were close to the same size as an eggshell, so that would be like our shot glass and our small cube. And then we tested some other vessels that were a lot larger, so that would be like the wayboat and the THP. And then we have some vessels that are kind of in the middle of that. And we so then that can all be seen in the table that's on the left, and then moving over to all the images on the right. So on the top there you have the eggshell. And then below that is the THP vessel that you can see is a lot wider. And then also the way boat, a lot wider again. And then back up to the top on the right, we've got 2 images of our small cube, so it's a little bit closer to an eggshell size. And then we've got the shot glass and the tumbler cup, which are close to the eggshell size and then also just a little bit bigger. So a little bit of like a medium ground there. So then our results that we saw with this is that ventral body wall deformation was showing up regardless of the vessel opening. So both of those chicks that you can see in those images, they both have different ventral body wall deformations. So then looking over at the graph that's over there, so the yellow bars that are in the top show embryos that didn't present with the deformation, and the blue bars in the bottom show embryos that did present with the deformation. So as you can see, all different types of vessels show ventral body wall, but then In-Ovo ones do not. So there's some difference in there that is causing the In-Ovo ones to not show that, and something with our vessels is causing this. Something that kind of complicates our results that we found is that we realized that temperature was changing in our incubator. So it wasn't maintaining at the level that we wanted to, so it wasn't staying, it was fluctuating above and below that. So that leads us to say that this hypothesis is inconclusive, because we don't know if what we were seeing with the ventral body wall was due to the vessel shape or if it was due to the temperature fluctuations. So this is something that we have to kind of circle back around to in the future.
All right, so after we started testing vessel shape, we also started looking at turning, which is another kind of difference between how eggs are naturally developed and how we're developing them. So hens, when they're like brooding and taking care of their eggs, they're actually flipping their eggs for the first 18 days of development multiple times a day. So we thought that this difference might be something worth looking into. And we found that turning eggs is actually helping to distribute electrolytes, nutrients, and water. So in this right figure, we are wanting these nutrients to move into like the sac that's like surrounding the embryo, and it's called the subembryonic fluid. So the nutrients in the yolk need to passively diffuse into this SCF fluid and get to the embryo. So we thought that if we start turning our embryos, then they might be getting more of the key nutrients that they need, and that might help prevent ventral body wall deformation. So to do this, we actually just Velcroed our different vessels onto an egg turner, which is kind of the figure on the left, and we basically just turn them throughout their incubation. Just like in the last one, we were having issues with temperature and humidity, so our data so far is inconclusive, and it's another thing that we're probably going to have to circle back to.
Yeah, so as mentioned in these last few hypotheses, we were having a lot of trouble with temperature throughout our incubations and chick development. So our optimal parameters, just to give you an overall view, is 37.5 to 37.9 Celsius with a relative humidity of 60 to 65%. And so we were thinking that because these temperatures were fluctuating outside that range, it was putting a lot of stress on the embryo and causing that ventral body wall deformation. So as you can see in the figure on the right there, the dotted green line is our optimal temperature parameter. And then you can kind of follow along, but obviously you can see that all of our different incubators were having a lot of temperature fluctuations. And so because of that unstable temperatures, we hypothesized that that was what was causing the ventral body walls, and why we are inconclusive in some of our other hypotheses.
So then getting to some of the overall conclusions that we've been able to obtain throughout the project. The first one, as stated, we have definitively disproved hypothesis 1. Again, it was believed that the ethanol used for vessel sterilization was causing the observed ventral body wall deformity. And within the experiment, we treated chicks with 10, 20, and 30% ethanol and compared the results against control chicks, which had no ethanol treatment. And then what we had saw was that both the treated chicks and controlled chicks, which were grown ex ovo, were seen to have the observed ventral body wall deformity. And with this, we were able to come to a conclusion that ventral body wall deformity being seen within the chicks is independent of ethanol.
And then once again, our vessel turning and our temperature hypotheses are so far inconclusive, so we're going to keep testing them. We were mostly just having trouble with the vessel turning hypothesis because there was such a fluctuation in temperature, and we want to be able to maintain other parameters before we start definitively saying if the vessel turning hypothesis was inconclusive. And we'll do more temperature testing later on this semester. We were mostly just monitoring it closely, but next semester I think we'll start paying more close attention to it. So last semester, fall 2019, we also hatched some chicks. So we do a lot of hard work, but we do some fun stuff too. So we ended up hatching 4 chicks. They were up in the research lab in the fall, and these were them.
So just kind of touching on our future plans, as Margot had mentioned earlier in the presentation, we're looking to kind of change our incubation time to around 65 hours to hopefully reduce the ruptures and potentially get some more eggs developing ex ovo. Another thing that we're looking to do is growing chicks in ovo, so continuing to look at what might be causing this ventral body wall deformation, kind of retracing our steps. In past semesters, they were seeing a few ventral body walls, but not that many, whereas now we're seeing almost 100%. So if we grow these chicks in ovo, we can see if we're the ones inducing the ventral body wall or if it's something else. And then finally, just continuing to test other hypotheses that were inconclusive, like turning, and also just continuing to look to maintain the optimal temperatures and humidity.
All right, so then these are some of our questions that we have for Bio 120 students, just as a way that you guys can get your extra credit points.